to set the stage around the efficiency theme that we want to tackle today. Bob, I'll start with you since you're big on my screen. So we've ascertained, that there's not always a straight line between the discovery of a potentially therapeutic molecule and the clinic. We're open to multiple takers and several different approaches. Why is it so important? Why is efficiency so important? Why is it important that we seek the straightening of that line, so to speak, in terms of the real time and resources that we're putting behind discovery?
Well, it's definitely much more cost effective, but you think about, the patient and getting the therapies to the patient as quickly as possible. That is the real goal that, Nadim and I will both talk about, like within everything that we do within the biotech and pharmaceutical industry, is getting there as quickly as possible. And that's where, trying to streamline and to get to more efficient processes really can help with that. And we're going to start talking a little bit more about artificial intelligence. But, as we think about how we're expanding into that space, one of the things that's very exciting to me is not just the efficiency gains that you can get to, like in terms of like the biologic process right now at this point, making a biologic drug, we've cut that timeline down to a third of what it used to be. So it's really a very significant amount of savings in terms of time. But the other component, as we start to think about moving forward, looking at different modalities that we wouldn't have access to before. So that's one of the things that is, to me, is very exciting about where we're going in this space, is really getting to essentially undiscovered country in terms of moving towards like degrader technology, and then maybe moving targets together. We have BiTE technology that we use within Amgen to do those things, to kind of bring in T-cell engagers and things. And that world is going to open up even more with artificial intelligence. I think we're on the horizon of a very exciting time.
Nadim, I want to pose that question to you and get your perspective. Cullinan being a biotech, as you said, you've got a phase 3 candidate, but multiple candidates in phase 1, sort of still having that startup biotech mentality. Do you have anything additional to add, sort of the I guess, importance of the efficiency play in discovery?
Sure. Yeah, I'll certainly start out by 100% agreeing with Bob that the why has to be centered around the patient, and the more quickly and efficiently we can move the process, more patients can benefit from our research. So I mean, that's fundamental, and so fully agree with Bob there. I think the more complicated answer is in the how, and Bob addressed some of that also. So I'll just give you a quick example from our pipeline to illustrate this. So for example, we know that CD19 now is a clinically and commercially validated target in B-cell lymphoma, and that there's multiple modalities pursuing these spaces in lymphoma. So with our molecule, CLN-978, which is a T-cell engager targeting CD19 and CD3, we recognize we were not first in class. And as I said earlier, we kind of focus on either first in class or best in class. So then we said, well, how can we get a best in class play here? So we actually designed a unique molecule to target CD19. And that was to create a fully human half-life extended T-cell engager, which targets CD19 with very, very high affinity. And we were able to show in preclinical experiments that we were able to have very efficient killing of CD19 tumor cells, often cells that would be considered CD19 negative. So that gave us an opportunity to take a potential best-in-class molecule. So it has 2 targeting domains, as I mentioned, CD19 with high affinity to kill low levels of tumor cells expressing that antigen. And the other targeting domain is CD3 on T cells, which then allows CLN-978 to redirect and activate T cells to seek out and destroy cancer cells that express the target antigen CD19. And so for me, this example kind of illustrates the question you were asking, at least the how aspect of that, Matt. It was, our starting point here, going back to what Bob was saying earlier, is it started with patient need. So we knew CD19 was a really important target, but then we pursued CD19 in a very different way by being creative in applying the right modality to that target. And so when we began our discovery and development of CLN-978 seemed to be a fairly straight line in addressing patients with B-cell lymphoma, a well-trodden route where we were trying to be best in class. But since then, we've seen that CD19 has really become an exciting target for autoimmune diseases, especially since the University of Erlangen group from Germany demonstrated deep and durable remissions of CD19 cell therapy in lupus, refractory lupus patients. So this was a really interesting finding. And then they followed up in December of last year at the American Society of Hematology, showing those deep and durable remissions extending into other autoimmune disorders. So we believe now that CLN-978 could potentially offer a potent, off-the-shelf, more patient-convenient alternative to CAR T therapy for patients with autoimmune disorders. So you could say that this was one where the path was originally straight, but sometimes discovery can lead to more than one path. So we're super excited about this opportunity.